Hello, good afternoon. Today I'm going to talk about cytosine base editor, CBE for short. I'm gonna first briefly introduce the concept about CRISPR-Cas9 before entering my topic that includes something like history and application. Here we go. In last year, 2020, the Nobel Prize in Chemistry has been promulgated to these two fantastic women, Emmanuel Charpentier and Jennifer Doudna. The Royal Swedish Academy of Sciences have complimented their feat as had a revolutionary impact on the life sciences, is contributing to new cancer therapies, and may make the dream of curing inherited disease come true. Indeed, the discovery of CRISPR-Cas9 is fascinating, which has boosted the development of a method for genome editing. Let's talk about the basic concept for CRISPR-Cas9 system. CRISPR Fulton is cluster of regularly interspaced short palindromic repeats, and Cas9 stands for the enzyme that cut foreign DNA using RNA with short sequence. The way it works is relatively simple. Like the picture has illustrated, Cas9 with guide RNA is first injected into targeted embryo. Then, like having the treasure map trying to find the fortunes, Cas9 begins to search for corresponding DNA compared with the inserted guide RNA. Once it's found it would unwind the DNA helix, snips the DNA, and subsequently breaks both strands. Because of this big and obvious break, the intruded cell will ultimately spot the problem and repair so that the new healthy part could substitute the viral part. Back to the Fulton for CRISPR. Cluster of regularly interspaced short palindromic repeats. Literally, there are two main points, repeat and spaces. In fact, the presence of nucleotide repeats and spaces are the trait to identify them from other systems. Consider the word palindromic. The first strand will have the same order as the second complementary DNA strand. Then graphically, you'll have these repeats which are identical, one after another, after another, after another. But as you can see from the illustration and from the names, they are interspaced by spaces, which interestingly are unique for each segment. And far ahead, there are Cas genes, which I've introduced, contain helicase and nucleus in charge of unwinding and cutting DNA. It is the fundamental structure of the CRISPR-Cas9 system. Before actually we present real application and how wonderful is it, I would like to spend like 3 to 5 minutes just focusing on history, the progress it has experienced. In 1987, Professor Yuzitsumi Ishino identified the CRISPR in E. coli when he was conducting a lab analyzing a gene responsible for the conversion of alkaline phosphatase and accidentally cloned a series of repeated sequences. However, due to the lack of the data and evidence, it is fairly difficult for these researchers to unravel the mystery of the errors. Not until 1993, the time Emden and his team discovered the CRISPR in tuberculosis and subsequently found the variation in the species in different positions. Around the same period, Francisco Mojica, the knowledgeable man over here, named this characteristic as CRISPR. In early 2000s, Mojica and co-worker Rod Jensen found that the spatial sequence is similar to the sequence found in bacteriophages, viruses, and plasmids, which was groundbreaking discovery. In 2012, Jennifer Doudna, Emmanuel Charpentier, and Fen Zhang proved the possibility to utilize it as genome editing tool. The achievement won the Nobel Prize in Chemistry last year. Beyond that, in the future, scientists have targeted at its bright potentials, including the development of epigenome editing, attempting to combine techniques such as transcriptional reprogramming. In case someone is intrigued by the history part, here is a more precise version. I don't think there's much I could comment or add on. But there is a small point that I would like to indicate. Although the technology sheds light on 2012, it has been further analyzed until 2015, when its first application had been popularized to the market, the virus-resistant tomato plant. The technology wasn't built by magic. It took so long time for the public to accept, and what I want to point out is that there is still a spacious room for development. Yeah, it has been granted a patent in 2017. So let's just hope it won't be another monopolized company in the future, just like insulin. Next part is about the current application. Thanks to previous report, it is way easier for me to understand what's going on here. I did cystic fibrosis as my topic last time. This is a disease that will produce thick mucus that clog the lungs and obstruct the pancreas. It is mutated because of a gene called cystic fibrosis transmembrane conducting regulator. For a specific type of this illness, class 2 to be more precise, CRISPR could recombine and collect the mutant F508del in intestinal stem cells and thereby eliminating the problem. It sounds promising, but current research fails to study the pathophysiology of cystic fibrosis in the lung, self-explaining the reason why there isn't any effective treatment for now. Next one is sickle cell anemia. It is also a genetic disorder because of the mutant protein. It has been deprived of oxygen carrying capacity and red blood cell malfunction. Fortunately, scientists from Vertex Pharmaceutical in Boston and CRISPR Therapeutics in Cambridge have validated the possibility to change region of a gene called BCL11A. And by changing it, is it possible to transform the fatal red blood cell into working healthy hemoglobin? It sounds brilliant, just like the treatment for cystic fibrosis, but it also has drawbacks as well. Firstly, it is tremendously risky. It could lead to other unexpected or unwanted consequences if something goes wrong during the procedure. The sample size, furthermore, is minute. Only 19 samples have been collected by Vertex, and side effect remains unclear. In general, there are three potential problems facing by CRISPR. Here comes off-target effects. No matter how precise you are, under current technology, sometimes the correction is accompanied by non-specific and unintended genetic modifications. Maybe you would like to fix things here, but accidentally you might bring influence to this area, this place, and this section. The consequences are serious and could not be ignored. Potential outcomes include tumor in animals and abnormal phenotypic expression in plants. Neither of them are wish and will be willing to embrace. Second problem is the DNA damage in DBS. CRISPR has to break both strands in order to fix it, but recent research have found that the repair of double-strand breaks seems to be imperfect. For mouse embryonic stem cells, hematopoietic progenitor, and human differentiated cell line, they all show large deletions and more complex genomic rearrangement at the targeted sites. In other words, the injury of DNA appears in certain situations, which scientists haven't figured out the condition. This undoubtedly increases the risk of the technique. Then it's about pleiotrophy. This word means a single gene could have multiple traits in different status, or in more simple way, a gene presence of deletion has more than one effect inside human body. No one could 100% guarantee that the corrected gene could last forever without turning into something else and express unexpected functions. Assuming you want trait A and you indeed succeed in transforming it, you could not ensure that it is internal without changing into trait B or trait C. The uncertainty is strongly linked with ethical problem. Whether it is moral to inject into human body with such high risk, I think nobody could identify an acceptable answer. If there is any method that could perform like CRISPR while minimizing this problem, the answer is positive. Base editor might resolve the issue if we exclude the last one, the moral stuff out of the discussion. Another explanation begins. Base editor had two classes, CBE, cytosine base editor, and ABE, adenine base editor. They mediate a C to T change and A to G change, respectively. Instead of breaking a large section of DNA, they center at a single base, or so-called homology-directed repair. If something goes wrong with, for example, cytosine on the illustration, here, it will first change into uracil on 3 strand and later makes change into A and T pair without affecting other elements. This molecular machine constitutes 3 main parts: guided RNA and Cas9 is similar to CRISPR. They will search for the intended portion, and cytidine dimelinase on the bottom right corner is in charge of converting C base into another base that acts like T base. UGI on the left-hand side, moreover, prevents base excision repair, which changes the U base back to the C mutation. This is pretty much it. I will give detail about the mechanism later, but first let me impart how important it is. I list 3 reasons this time. There are more actually, but these 3 are typical. First one is apparent to clarify the effect of a certain base mutation on gene function. As we all know, both CBA and ABE target on a single base, which work as a fantastic tool to activate the influence of small, seemingly insignificant mutation. Not to mention that around 2/3 of the human diseases are related to single nucleotide variant. Rather than applying traditional CRISPR technique, it would be better to use base editor to figure out the pathogenesis and corresponding treatment. For the third perspective, single nucleotide is related to many genetic bases of phenotypic variation for animals and plants. By learning it, we could actually promote more product to the public with faster pace than CRISPR, which released the antivirus tomato 2 years after the introduction of the technique, helping both agriculture and stock farming to develop. How exactly does it work? The first step is the same as CRISPR. The system is injected into the cell and will find the targeted place where it should be fixed and form the protein RNA DNA ternary R loop complex. Then it is cytidine dimelinase, the red part over here, turn to induce some base on non-complementary strand from C base to U base, or more precisely, deaminated the amine into oxygen atom. Because uracil doesn't exist in DNA, after DNA replication or repair, U base turns into T base, pairing with A base, so you will finally get a base edited DNA reversing the incorrect base pair. I put it here with some explanation just in case someone who could not access my video or my presentation could learn the idea by scanning through the text. Basically, this is the same as the previous slide. Again, by comparison, there are lots of advantages. First thing is that it does not involve double strand breaks, so that it avoids the possibility of non-homologous end joining. The pathway sometimes leads to loss of nucleotide or even incompatible overhangs. Second thing is like the illustration, insertion, deletion, and friendship won't happen with CBE. Therefore, avoid the possibility of randomly inserting unintentional editing. In fact, CBE has been praised by scientists as new gene editing technique with higher precision. Indeed, lots of versions have been testified since the invention of this technique. The first generation CBE1 used AppleBake1, an enzyme from rat, and that Cas9 system as the ingredients. Because of the UDG, the element that could reverse the situation back to the original one, the base additives efficiency is very low, in the range from 0.8% to 7.7%. Second generation integrates UGI into the system, raising the efficiency to around 20%. In order to raise the conversion rate, scientists substitute the dead Cas9 cell with the living one, so that you could cut and snip a notch in the non-complementary strand, meaning UG pair will first transform into UA pair, then become TA pair after DNA replication. It succeeded in raising the efficiency to around 37%. The fourth generation adds a second copy of the UNG inhibitor, UGI, increasing base editing product purity so that minimize the effect from UDG. The efficiency of for this model is more than 50%. Compared with CRISPR with maximum 31% of efficiency, the fourth generation is fairly high and reliable. However, even with these numerous advantages, there are still unsolved problems. The off-target effect could not be eradicated. Maybe the base editor has torn bigger section than expected, so that nearby base has been influenced or mutated from C base into T base. There are still rare chance for DNA deletion and rearrangement to happen, unfortunately, since scientists haven't mastered in genetic editing yet. So in conclusion, CBE targets at homology-directed repair mediated C base to T base without double-strand break. CRISPR-Cas9 targets on a specific sequence and would repair the cell by breaking both strands. The invention of CBE kind of resolved the low editing efficiency issue of CRISPR-Cas9 for HDR. And this is the end of my presentation. Thanks for listening. These two slides are my reference.